This is the advanced manual for the iFL. It is highly recommended that you view the basic manual first before proceeding. We're going to cover measurement of RD, respiration in the daytime, gamma star, the compensation point where there's a lack of mitochondrial respiration, GM or mesophyll conductance, and how to make ACC curves, photosynthetic rate as opposed to CO2 at the site of carboxylation. In the making of ACC curves and measurement of GM, it's important to also measure chamber leakage and correct for it for reliable results. ACI curves compare photosynthetic rate to CO2 in the intracellular air spaces, or CI. ACC curves compare photosynthetic rate to CO2 levels in the chloroplast at the site of carboxylation in the chloroplast stroma. So GM, or mesophyll conductance, is affected by its species, resistance of the cell wall, the cell membrane, the cytoplasm, chloroplast diffusion, temperature, and CO2 level. CO2 in the intracellular air spaces has to diffuse through the cell walls and membranes, cytoplasm, chloroplast membranes, and in so doing it runs into variable resistance. However, because we're talking about fluxes, it makes more sense to talk about uptake. Therefore, GM is used to measure mesophyll conductance, not resistance. So CC, CO2 at the site of carboxylation in the chloroplast, is equal to CI, the CO2 level in the intracellular air spaces, minus A, or photosynthetic rate, divided by GM, or mesophyll conductance. So why measure GM? Well, there's a strong consensus among the leaders in the field that ACC curves should be used for measuring photosynthesis parameters instead of ACI. Why? Well, it's been found that ACI curves are not as reliable as ACC curves to characterize plant activity, especially in C3 plants. Instrument chamber leakage is also more of an issue with ACI curves rather than ACC curves, because A and CI values move in different directions due to chamber leakage at different CO2 levels, while A and CC values move in the same direction due to chamber leakage. There are issues with GM. In C3 plants, it's been found that as the temperature rises from 10 degrees centigrade to 40 degrees centigrade, GM changes. And if you look in the upper right-hand corner, you'll see a GM temperature curve. GM also changes at high actinic light levels. And GM changes under drought stress and salt stress. This is a comparison of an ACI curve versus an ACC curve on the same leaf. Notice that the ACC curve is in red and the ACI curve is in black. There are two different equations that are used for determination of GM, or mesophyll conductance. One is the variable J method, and the other is the constant J method. The variable J method is recommended for the LASK protocol in C3 plants. It's used when J does vary, or electron transport rate does vary if CO2 levels change. Usually it's, again, below ambient. The constant J method is used where electron transport rate does not vary much if CO2 levels change, commonly at higher CO2 levels. RD is respiration in the day or mitochondrial respiration, and gamma star is a compensation point or point where there is an absence of day respiration. The variable J method. J is electron transport rate, usually from chlorophyll fluorescence. When J does vary, if CO2 levels are changed, usually it's used where CO2 levels are below ambient. It may be used above ambient, however, it has usually been used where J was variable. It is common to use the method with an air tank that has a 1 or 2% oxygen level, but not 0%. This is done to reduce photorespiration. It's more sensitive to gamma star and RD than other methods. Values are usually lower than the constant J method. And A, or photosynthetic rate, are limited by the enzyme Rubisco. The constant J method. It's commonly used where J does not vary much if CO2 levels change at higher CO2 levels. Usually, it's used at CO2 levels above ambient. J should be below saturation for best results. A, or photosynthetic rate, is limited by RUBP, and the method is sensitive to gamma star variation. When measuring GM, you have to deal with the fact that GM changes with temperature. A simplified way to deal with this is the von Kamer constant temperature correction. Gamma star von C is equal to gamma star Lask plus RD Lask divided by GM determined by the Lask method. Now, as I said, this is a simplified way to handle temperature. However, most experts prefer to measure gamma star at all temperatures that are relevant to an experiment for best results. There are different ways to measure GM. Some are preferred for C3 plants, some are preferred for C4 plants. The most common way to measure GM in C3 plants is the Lask protocol, and it's considered authoritative by many for C3 plants. It is not recommended for C4 plants. The Koch protocol for RD is very commonly used for C4 plants. Gamma star is estimated and not measured. RD is measured. It may be used for C3 plants. RD is higher with the Koch protocol. The Yin method is a newer method that measures RD, and gamma star is also estimated. Those three protocols are available on the iFL. Another method for measuring GM is the carbon isotope method. It's used by many experts. It requires an additional large chamber without seals in the leaf, and you take plants grown in a C12 environment and put them in a C13 environment. This is used by people that can afford it. It's an expensive approach, and the carbon isotope method requires specialized equipment. RD, again, is respiration of the day, and gamma star is the compensation point, or lack of respiration. This is an iFL screen for the setup of a Lask protocol used for C3 plants. The LASIK method uses 3 ACI curves. However, unlike normal ACI curves, all 3 actinic levels should be below saturation levels. It's commonly done at 300 micromoles, 150 micromoles, and 75 micromoles for sun-grown leaves and shade leaves. Indoor plants will have lower values. These are the common steps for making a LASIK protocol. You're going to have a range of CO2 levels from ambient down to low levels, and you're going to have light levels that are in the right-hand column. Each step should be 2 to 7 minutes for each CO2 level. It takes longer for sun-grown leaves to stabilize. Shade plants adjust more quickly. You start with the highest actinic light level first, and the actinic light levels are always below light saturation. This is a screen for post-processing on the iFL for the LASIK protocol. It's difficult to measure a LASIK protocol in the field due to the time requirements and temperature variation, because of course GM does vary with temperature. Number 2, it's common for 3 or 4 lines from the LASIK protocols, the ACI curves, not to cross exactly at the same point. The instrument uses an algorithm, and you can see the curved red line on the screen that shows the algorithm attempting to find the nearest point of coincidence to find where gamma stars should be. Sometimes leaves become inactive under clamp due to drying of the leaf. One can overcome this issue by either increasing the flow rate or using the wetter column to maintain near ambient conditions. Chamber leakage is an issue for the LASIK protocol because you're dealing with low light levels and low CO2 levels. So leakage is a significant issue. To get around this problem, we included the Flexus chamber leakage protocol. And what Flexus did is he would take a leaf of the type that is used in the experiment and boil it. Then he would check to see that the leaf was dead by trying to measure chlorophyll fluorescence. If there was no variable chlorophyll fluorescence, then he's sure that the leaf is dead. After that, he would put the dead boiled leaf in the instrument measuring chamber and do an ACI curve. And the corrections for leakage then can be transferred to LASIK protocols or other experiments moving forward. This is an iFL graph of the Flexus chamber leakage protocol. Taking a dead boiled leaf and putting it into the chamber and then doing an ACI curve on the leaf. Notice that the green line represents photosynthetic rate A and the blue line represents CI. The values are then input into the LASIK protocol or other measurements to correct for any chamber leakage, reducing chamber leakage to the minimum. This is the post-processing graph for the Koch protocol used for C4 plants. Issues and limitations of the Koch protocol. It's recommended for C4 plants. It will work on C3 plants. RD is higher than LASIK values in the Koch protocol. This is an AQ curve, or A versus low light level curve. Gamma star is calculated, not measured. It will work for plants grown at any CO2 level or temperature. It assumes GM is infinite, but it is not. And the stoma may close as PAR levels for Q approach zero. These are the steps that are commonly used for the Koch protocol. The curve is A, or photosynthetic rate, versus light level, or PAR. 400 micromoles, 300 micromoles, 200, 150, 100, 75. The last one is at zero. Each step should take 20 to 30 minutes because we are using a white light source. This is the post-processing graph on the iFL for the Yin protocol, which instead of A versus PAR, like the Koch protocol, is A versus J, or electron transport rate. Gamma star is calculated. The Yin protocol. The Yin protocol uses the same approach as the Koch protocol, but instead uses electron transport rate in measurements from chlorophyll fluorescence. Light levels and step times are the same as with the Koch protocol. The Yin protocol finds that A, or photosynthetic rate efficiency, decreases as actinic light level increases, while the Koch method assumes that photosynthetic rate efficiency remains the same. RD is lower with Yin than with the Koch protocol. This is an iFL graphic representation of FM prime correction called Multiflash. The FM prime correction capability does not need to be used for the protocols that we've just discussed. It doesn't have to be used with the laced protocol because you're using light levels that are below saturation. The Koch protocol, the same thing, and the Yin protocol, the same thing. Square top flashes work just fine. The iFL uses a large area infrared leaf temperature sensor underneath the leaf. It provides a more reliable measurement because it's averaging over a large area. Now, there are conditions that lead to temperature heterogeneity, and this reduces that as a problem. If the leaf is too small to use with the IR leaf temperature sensor, you have the choice of either using a thermistor that plugs into the side and measuring leaf temperature that way, or calculating it. This is a picture of the measuring chamber of the iFL, and you'll note the round sensor is the leaf temperature sensor. It covers about 70% of the measuring chamber. This is a picture of the wetter column mounted on the iFL. It allows you to program in near ambient humidity conditions or above ambient humidity conditions. It's great for ACI curves. The chemistry of the wetter column. Iron 2 sulfate 7 hydrate becomes another chemical as it gives off water. So it starts off as melantorite, and it dehydrates into either siderotyl or rosenite, giving off only water. The column is blue when it has fresh chemicals in it, and it changes color to purple when it's spent. General information. Many of the instrument housekeeping functions have been automated to eliminate or reduce errors and make the instrument easier to use and allow walkaway automation. Gas exchange automation includes the Erga Auto Zero, which corrects for drift for every measurement. The wetter column helps prevent samples from becoming inactive due to instrument flow rate dehydration. Unlike H2O ergas, the iFL has solid state H2O sensors that do not drift over short periods of time, do not eat lots of battery power. And as far as chlorophyll fluorescence automation, the instrument has an automated modulated light setup to eliminate possible errors. When doing chlorophyll fluorescence, if the saturation pulse is too wide, in most instruments, you get something called saturation pulse NPQ, which averages the curved portion of the saturation flash that is caused by the saturation flash being too long. The iFL eliminates this from being a problem by using an 8.25 millisecond rolling average that finds the highest saturation flash intensity value and eliminates saturation pulse NPQ from being a problem. The instrument also has FM prime correction for when near saturating light conditions are necessary. Measuring files. The files in the iFL are comma delineated, and so you can open up the measuring files using the data card, putting the data card into the USB adapter and putting the USB adapter into your computer. The files will open in Excel or other spreadsheet systems. Checklist for ACC curves using gas exchange and chlorophyll fluorescence measurements. When using the LACE protocol, it is recommended to use the Flexus chamber leakage protocol to correct for CO2 leakage. When done, engage the correction. The von Kamer gamma star correction can be used for temperature correction. However, it is always better to perform the LACE protocol at different temperatures that you will use during your experiment. GM, RD, gamma star all vary with temperature. When measuring RD and gamma star using the LACE protocol, no par value should be near saturation. Number 4, usually there is no exact point of coincidence for all three ACI curves when using the LACE protocol. As a result, the iFL uses a mathematical algorithm to determine the nearest point of coincidence of all three curves. It is this calculated point of coincidence that determines gamma star on the x-axis. Number 5, the instrument will provide direct readout of GM and CC for ACC curves. However, EC max and J max must be calculated using separate software.